Welcome. So these are the first 3 of 6 sections of this chapter. The next 3 sections, the second half of chapter 25, is a little bit shorter than the first half. I guess I probably shouldn't call them halves, but a lot of the information is front end loaded into this chapter. So we've talked about, in general, what the idea of evolution through natural selection is. We've talked about microevolution, where the gene frequencies in a population can change, either randomly or because nature is selecting for or against certain genes and traits. And then with chapter 24, we talk about more macroevolution, which is how a population can get isolated, start changing into its own form. And we ended that chapter with a little blurb on speciation, which is a population becomes isolated for so long, changes so much, it becomes its own new, unique living organism. Chapter 25 is basically a history lesson, and it covers 4.6 billion years of history. So real quick, just get through it like that. We have one more chapter, which we will try to get in next week, so that the week after next, we can review and take the test before spring break. So to start out with, we have a discussion of where did you get the first living thing? All right. So it's, from a scientific perspective, pretty obvious that the first living thing had to arise from non-living sources. So how would that come about? Well, these are kind of your 4 major requirements, really, I guess, on any planet, that would have to happen in order to get to the first single cell that can then reproduce and turn into 2 cells, and over about 3.5 billion years to 4 billion years diversify out into the life we see today. So we're going to talk about each of these, forming small organic molecules, those polymerizing into big organic molecules, where your first kind of membrane-like structures would come from, and then how do you get the first signs of sustainability of such a system, reproduction in some ways, but what we would call self-replicating molecules. So the next few slides, we're going to have 4 or 5 slides that go through each of these points, and I'm going to try to talk about them in a little bit more specificity. Okay, so name me some of the basic organic compounds or molecules that we talked about like in Unit 1 a long, long time ago. What's a basic organic molecule, biomolecule?
Phospholipids.
Yeah, your lipids. Your lipids, which are made up of like glycerols and fatty acids.
You ready for this?
Okay, I'm ready.
Dihydrogen monoxide.
Dihydrogen monoxide, aka water, is actually, I'm no organic chemist, but I don't believe it's an organic molecule. However, it is certainly, and from what we can tell, a necessary component for starting life.
You're right. So like methane or...
Yeah, methane is probably going to be your simplest organic molecule, just a carbon with 4 hydrogens surrounding it. But then we talked about like amino acids, nucleotides, saccharides, like monosaccharides or polysaccharides.
Sugars.
Which are your sugars, or sugars are within that group. Okay, so There's some background information. I'm not going to go through every point, but from what we can tell, and from our piecing together of the deep history of Earth, it did not start out like what you see around you. We didn't start out with oxygen in the atmosphere, which is probably good because oxygen is pretty harsh on organic molecules and living things that aren't kind of evolved to use it. And there were other gases. Methane, ammonia is thought to be pretty common. A very humid planet, lots of water vapor probably existed on Earth. Not exactly 4.6 billion years ago, but 4 billion years ago. The early part of Earth's history, it was a molten ball of rock. I mean, it was so hot that you just had fluid rock that made up the surface. It took a while for that to cool down enough to where it wouldn't just sterilize any kind of organic matter or living things that cropped up. But the ancient Earth was very different, with a different set of chemicals available. Okay, so that's the main idea here. And we have, and I'll go into this a little bit more later, we have some pretty classic examples of experiments like the Miller-Urey experiment. I think that was Unit 1 where I mentioned that. Anybody remember that name? The Miller and Urey experiment?
Yes, sir.
Yeah, okay. Are you just saying yes to cheer me up on a Friday?
Isn't that dude who made like a mini environment within a lab that made organic molecules?
Exactly. And Miller and Urey, I know I just said that person, but it was two people that they created essentially what they thought were the conditions of ancient Earth with water vapor, carbon dioxide, methane. They had electrical sparks to simulate what we think was probably a pretty violent climate with lots of intense kind of electrical storms. And in the lab, we have tried to simulate these conditions, and when we do, we make amino acids and nucleotides, amongst other organic kind of components. So the basic building blocks we have shown in a lab, you can make them from non-living sources. Now, how do you get from there to macromolecules, to larger molecules? Well, as I just mentioned, we have seen, and I like this terminology, spontaneous abiotic synthesis. Spontaneous just kind of means it happens on its own. What does abiotic mean?
Not living.
Non-living, not living formation of these different small components, basic building blocks of life. Now, what we have found is that if you give them some sort of substrate, and they mentioned sand and rock here, I'm not familiar with that, but I know they've done experiments with clay. Some types of clay, and I don't mean like silly putty, I mean like kind of the red dirt that you see around the southeast. That is soil that is made up of very tiny particles, and when they come together, they make kind of a matrix. And that matrix, that surface that clay in particular can make, it almost acts like a workbench, a chemistry workbench. Clay seems to encourage these tiny organic molecules to stick to the surface of the clay, and then they could stay there and chemically interact with each other. So we have also seen in the lab where you could get, say, nucleotides. They talk about the RNA monomers. That's A, U, C, and G, adenine, uracil, cytosine, guanine. We can form these in the lab, and then when they are dripped onto some specific surface, not every surface works, but on some surfaces like clay and apparently sand and rock, you can begin seeing them polymerize. They will bond together and make chains, polymers of these smaller components. And there you are. You're at a point where you're making then bigger macromolecules from these smaller organic building blocks. So the chemistry of life seems to be possible without any life present. Now, the next step to getting towards the first functioning cell is what we call protocells. Proto kind of means early or before. So you have to get some kind of cell-like structure. And when we say cell-like, we're really referring to a general like sphere, a membrane structure of some kind. Well, this doesn't take much high-level experimentation. If you put, there are a lot of lipids. Phospholipids, for instance, are one of these. If you put phospholipids into water, they will interact with each other and with the water, and they will form a bubble of phospholipids, which is basically what your cells are. It's a phospholipid bubble with a lot of things going on inside. So we can create these, what we call protocells, these kind of vesicle-like lipid bubbles in water pretty easily. And this is what they look like under a microscope. So you mix some phospholipids into water, and they will begin to form these bubbly structures. And honestly, I mean, that's almost what a cell looks like underwater. I mean, under a microscope, in water. Now, the importance of some sort of structure is that you're separating an internal environment from the external environment. And that's what your cell membranes do. I mean, that's why we talk about cell membranes a lot, because they are able to select what comes in and out and give a stable environment for your cells to function in. Again, they've tried to do this, and what they have found is with clay present, you get a much better kind of rate of production of these vesicles and of macromolecules in general than if you do not have clay or some kind of surface. Now, we, at this point, have a cell membrane-like structure that we can show forms under abiotic conditions, and a lot of the organic molecules. Where it starts to get tricky, and we have seen these things happen in a lab as well, self-replicating molecules. RNA is actually a pretty diverse class of chemical. And last unit, I didn't go into them very much, but we mentioned in your reading ribozymes. RNA actually has the ability to act kind of like an enzyme. It can help other organic molecules initiate certain chemical reactions. And they have found that RNA in a non-living setting can make copies of itself with just kind of basic base pairing, like A to U and C's to G's. They can kind of initiate the formation of other strands of RNA. So we think the first living things were probably an RNA RNA-based life form. In fact, the term is RNA life. That is the current kind of best model, best explanation that we have at this point, is that some sort of self-replicating RNA, a strand of RNA that could, under the right conditions, encourage a complementary strand of itself to be made, which under the right conditions could encourage another strand to be made. Once you start making copies of sequences, you start kind of creating information, which is very important to forming a life form. If that gets caught within a protocell, you now have a membrane-like structure with self-replicating properties, and it seems like, based on laboratory observations, that's as close as we can see to a living thing. How you go from self-replicating RNA inside a bubble of fat to whatever that first cell was, that if we ever were able to see it would go, that is life. That is a gap we have not filled in yet. So there are, this is hypothetical modeling that we're doing here, with a lot of experimentation backing it up. Okay, second section, which will go a little faster. In this section, they talk a lot about the fossil record. So I'm going to go into some basics about it. If it was a nicer day, and one day when it is nice, we might go outside. There is one of these layers written down here is actually right outside the window there as a rock layer. And what we've done in the field of geology is broken rocks around the world down into certain layers. Some of these words might look familiar to you, like what? Yeah, Jurassic Park is a pretty popular series of movies. There's the Jurassic right there. By the way, a lot of dinosaurs that are featured in the Jurassic Park movies were actually from the Cretaceous period. Not from the Jurassic, but whatever. You've probably seen words like Mesozoic, Pleistocene, maybe, when people talk about the woolly mammoths and things like that. But around the world, there are many different kind of rock layers that you can find. And usually they contain a very specific set of fossils. And so what geologists have done over the last 200 years or so is classify rock layers around the world into these different layers. Now, some of them have really weird names. A lot of them are named after the places where we first discovered the rock. They're not listed here, but in the Carboniferous, you actually have Pennsylvanian and Mississippian layers, which are named after those states. Cambridge, Devonshire. There's a lot of just places in here, so it might look a little weird at first. But every one of these geological layers, we have dated them to being a certain age, and we have categorized them based on fossils we find within them. Now, in northwest Georgia and on this campus, if you look again at that rock that you have out there that they cut into to build the science building, that is actually rock from the Ordovician time period. That's 450 million year old rock out there. All the rock from all these time periods since have actually just been eroded and washed away. When you're on a mountain or a hill, erosion is much more intense. If you go out to Montana and Utah in the western states, you will be able to find rock from Jurassic and Cretaceous times. It is still there in those locations. And in other locations, you can find much more recent rock layers. It's hard to study this. I think there's like one or two locations on Earth where you can look at every layer and none of it has been completely eroded away and disappeared. So in some parts of the world, you have certain layers of rock. In other parts of the world, you have other layers of rock. But based on studying all the rock layers around the Earth, we are able to put together a full picture of kind of the geologic history of life. You do not need to memorize any of these. I know you're probably worried about, like, well, what's going to be on the test? I might even just give you this chart and ask you one or two basic questions about, which layer is older. Something that simple. It's not that complex of an idea, even though it looks all complex. Now we have large periods of time classified, and then we break those down into what we call periods, and then we've even got what we call epochs in here, and we're currently in this top one, the Holocene. That's our modern one. How do we know these ages in millions of years? Well, the book talks about half-life and using radioactive isotopes. So we use what's called radiometric dating. You basically find some sort of element in the rock that at one time was radioactive, a radioactive isotope, which most elements have some kind of radioactive isotope that can exist. And then you look how much of that radioactive material is still left in the rock and how much of it has turned into its final decay product. If you've had AP chemistry, I know you've done this, right?
We haven't. Oh yeah, we have done this.
Maybe like just parent-daughter isotopes, things like that.
Yeah.
That's usually at the end of the curriculum.
Yeah. Oh, at the first semester at the end, is that what you said?
Wait, so you're talking about half-lives, right?
Yeah.
Yeah, roughly.
Okay. Sometimes
Like,
in regular
she
chemistry you
changes
get
it, she
into it.
changes it each year.
Yeah.
So,
I mean, we might cover it, we might not kind of topic in chemistry classes.
yeah.
But anyway, with half-life, radioactive materials work in this kind of weird way where there's kind of a set amount of time, and over the course of that time, half of your material gets converted from radioactive to the non-radioactive stable form. I don't want to go into this too much. Your book does a pretty good job of explaining it. But you can basically look at ratios of the radioactive to non-radioactive version and see how long would it have taken for that sample to look like that. And then you work backwards. And that's what we rely on for our kind of testing of the age of rocks. Now, what can we learn from it? Well, once you know a time scale, which is honestly typically deeper rock is older, rock near the surface is newer. That was the purpose of, part of the purpose of looking at the rock samples in the lab the other day. You can look at fossils that you find in later and more recent time periods, and thanks to understanding which layers are older and newer, and to some extent how old they are roughly, you are able to piece together a picture of how populations change. So in this case, we see kind of a pre-mammalian world, before the mammals, what a lot of the animal skulls look like at that time, and then how the skull changes, which specific bones change, and how you gradually go from, populations without mammals to the first mammals arising. By the way, a lot of mammalian change happened in the skull and teeth. That's a pretty important set of phenotypic changes that have occurred over the years. And we can also piece together these trees of life where we can kind of see what the populations look like and how they change. That's all recorded in the fossil record. And finally, We'll be right at 30 minutes here, like I promised. Section 3 of chapter 25 basically gives you a quick wrap up of billions of years. Now, there are a lot of things that you can look at up here, but here are the major events that life seems to have gone through over its course of time here. Your first prokaryotes, dealing with an oxygenized atmosphere, eukaryotes showing up, multicellular eukaryotes showing up, animals moving on the land, and then of course kind of present day, which they just label as humans. They do this kind of fun thing where they take the entire history of probably the earth, I guess. Yeah. Where you go from the origin of the earth all the way to right now, and they condense all of these events into 1 hour.
Wow.
And what's, I think, most fascinating about it, besides the fact that the first 15 minutes of that hour, we don't really think there was any life, or at least it was this like pre-life kind of stuff we were talking about at the beginning of the chapter. The evidence for humans would stretch back on this timeline 0.2 seconds out of that hour that the earth has been here. So again, if you convert the 4.5 billion years to an hour, it's 1/5 of 1 second that human life has been on this planet. We are an incredibly recent development based on what we see in the fossil record. And you don't have to go back very far. I mean, this is 500 million years ago. You get in a time machine and you go back a little bit more than 500 million years, you're walking around on just bare rock. No life was really even on land. And that's also fairly recent if you look at this timeline that stretches back, scaled out. I mean, just even life on land is pretty recent of a development. Okay, so I think the reading will be less terrible than it's been all year, unless you just totally dislike this kind of material and miss talking about transcription and translation. I'm going to stop there. And what I want to do today, instead of jumping ahead to some kind of lab activity, we've done 2 labs. One of them you need to get done today. If you need time to work on that variation in populations lab, you have it right now. If you are done with it, and when you are done with it, I am going to ask that you choose a living thing for your video project. I don't have to have that in like writing, but I would want you to tell me when you have chosen an animal or plant, if you wish. But what's going to be the topic of your evolutionary history project? And once you've chosen that topic, just let me know so that I can tell you, oh, that's a terrible one, or that's great. Sometimes people do things like, I'm not going to be able to think of a great example, maybe like, I want to look at the Luna moth. And I'm like, well, why don't we just do like moths and butterflies as a whole group? And sometimes people are like, I want to look at how reptiles evolved. Well, let's pick a more specific living thing. So that's what I want you to do with the time we have left. Get a foundational start to that project after you've turned in your variations in population lab or anything else that you need to turn in. So if you could stop the video, make sure you do all your reading this weekend. Stop the video.